Great. Yeah, thank you so much for the invitation. I'm excited to be here with you all today and tell you a little bit about what we've been working on. And yeah, it's been really great to hear from everyone in the earlier speeches in the day. Yeah, so today I am excited to share with you some work that we've been doing on better understanding the impact of gene count normalization methods on analyzing data from imaging-based spatially resolved transcriptomics technologies. Let's see. Yeah, so as we have talked about and discussed at length earlier, spatially resolved transcriptomics technologies can be categorized into two classes. Imaging-based SRTs rely on multiplexed in situ hybridization and are very high resolution, resolving single molecules in space. For these technologies, you have to design the capture probes based on the base sequence of the species you want to detect. And although these technologies have been demonstrated to work with up to several thousand different RNA species, as we've seen in the previous talk, typical experiments nowadays usually include a panel of about a few hundred genes. And in contrast, capture and sequencing-based methods rely on untargeted capture of RNAs, and they're generally lower resolution. And because of the untargeted nature of the RNA capture, they're able to capture the whole transcriptome. And so in the work I'll be presenting here, I'll specifically be exploring gene count normalization approaches with imaging-based spatially resolved transcriptomics data. As I'll be talking about in a little bit more detail later on, the smaller and targeted nature of the gene panels presents questions on whether or not commonly used normalization strategies will work as expected with this type of data. There's a lot we can do with spatially resolved gene expression data, as we've been talking about extensively today. To begin probing at biological questions and generating hypotheses from the data, there are some typical analyses that people commonly do, and that includes differential gene expression analysis between cell types of interest, tissue regions of interests, and that can be done with a few different statistical and computational methods, many of which are implemented in popular packages like Seurat or Scampy. Specific to spatial data, you can look for genes with significantly variable spatial variation using methods like meringue, NNSVG, spatial DE, amongst other popular methods. However, the first step before any of these statistical investigations is gene count normalization. And what I mean specifically here, I'm using normalization to refer to the adjustments made to the detected number of raw gene counts before any downstream analysis. And so today I'll briefly go over what the goals of count normalization are, what current normalization approaches exist, and how we're evaluating performance of normalization approaches. I'll go over a specific evaluation we did in one case study with merfish mouse brain data, and talk about how the results from that case study extend to different contexts. And then finally, I'll talk about some recommendations we suggest based on some of the observations we made in this study. And so to start off with, what are the goals of count normalization? Broadly speaking, count normalization aims to control for technical variation that may result in biases in downstream analysis. So let's say you're measuring 3 genes in 2 samples, and for some reason, some technical reason, your detection is way more efficient in sample 2 than in sample 1, and so you see proportionally more counts of all genes in sample 2 compared to sample 1. If you perform differential expression testing on this data, you'll find that all 3 genes are differentially expressed, but this is not actually reflective of any biological difference between the 2 samples. It's just purely due to the technical differences leading to the higher detection in sample 2. And so gene count normalization attempts to correct for this before any downstream analysis so that you don't have technical differences obscuring any interesting biological differences between your samples. And so gene count normalization has been applied to various gene expression data types. For bulk RNA sequencing, it corrects for things like stochastic variation in PCR efficiency, reagent variation. In single cell RNA sequencing, it can correct for stochastic RNA capture. And in imaging-based SRT, we know that not all cells are fully imaged in our data, as they can lie either above or below the planes that we're imaging, and that may motivate the need for similar gene count normalization approaches. There are a number of gene count normalization methods that have been developed over the years. Library size normalization simply scales the number of gene counts for a specific gene in a specific cell by the total number of counts detected in that cell. And this method has been applied to bulk RNA sequencing, single cell RNA sequencing, and has also been applied in some imaging-based SRT data. DE-seq2 and TMM, as implemented in the EDGE-R package, were developed for bulk RNA sequencing, and specifically tackle the question of compositional differences in gene expression and how they might confound simple library size normalization. SC-transform is a method specifically developed for single-cell RNA sequencing data, and it constructs a generalized linear model of gene counts, where library size is your predictor variable, and it then uses the residuals from the resulting negative binomial regression as the normalized gene expression in downstream analysis. And then finally, with imaging-based SRT, cell volume or cell area is sometimes used to scale detected gene counts to get normalized gene expression. And one thing to note here is that library size, DE-seq2, TMM, and SC-transform are all count-based normalization methods. They use some variation of the total detected counts as sort of a surrogate for the technical variables that we want to correct for. And so the first question we wanted to ask is, do we need to normalize gene counts at all in imaging-based SRT data? And so to answer this question, we started off with a simple simulation with two populations of cells of the same cell type that are on average in different locations in the vertical direction. The green population of cells is more or less centered within the Z-planes that are imaged in this hypothetical experiment, and the orange population sits kind of above the planes of imaging. Based on their positions, we compute how much of each cell's volume is captured and sample its simulated transcriptome according to how much of the cell volume is captured. And so on the right here, blue means that none of the cell volume is captured, and then the gradient up to red, where red is means that the entire cell volume is captured. We then run differential expression testing and see if we see any significantly differentially expressed genes. And because these cells are sampled from the same transcriptional distribution, we expect to see no differences between them on differential expression testing. However, when we run the differential expression on the simulation, if we don't normalize at all, we find that we have a very inflated type 1 error rate, where we identify many genes to be differentially expressed when we in fact know that there aren't any differentially expressed genes between those two populations. And this effect goes away with any normalization strategy. And so this indicates that, yes, it seems that normalization might indeed be necessary with imaging-based SRT data, and specifically the normalization is necessary to correct for the amount of the total cell volume captured in our imaging data. And so given this result, we set out to investigate the performance of gene count normalization approaches with imaging-based SRT data. And so to evaluate performance, we consider that imaging-based SRT data often has gene panels that are much smaller than the whole transcriptome. And often gene panels are designed to focus on a specific cell type or a specific region of interest, or even a specific biological process of interest. And ideally, if we use two different gene panels to assay the same tissue and run our downstream analysis of interest after gene count normalization, whether that analysis is differential expression testing or spatially variable gene detection, we should ideally reach the same biological conclusions regardless of the gene panel used. And so we designed a simulation study based on this thought. And so now I'll delve into detail about one of the simulations we ran, where we simulated a brain ventricle specific gene panel from MERFISH mouse brain data. And so the original MERFISH data measures 481 genes. We simulated gene panels skewed towards the ventricle region of the brain by randomly selecting 100 genes that are differentially overexpressed in the ventricle. We then normalized the gene counts in each panel using the 5 normalization methods that I mentioned before. And we performed several downstream analyses, including differential gene expression full change analysis, as well as spatially variable gene identification. And we compare the results we get with both panels for the 100 common genes between the 2 panels. And again, ideally here, we're looking for normalization methods where the results don't change when different panels are used. And so the first thing we look at is cell-specific scaling factors that the different methods used. And the scaling factors are the numbers that the gene counts in each cell are scaled by under these normalization methods. And so for cell volume normalization, cell volume computation here is not affected by the gene panel that you use. And so the cell volume computed with the full gene panel is exactly the same as that computed for the skewed gene panel. However, when we look at the scaling factors computed with the count-based normalization methods, we find that there's a significant tissue region-specific bias when we use the ventricle skewed gene panel. And so here I'm showing the scaling factors computed with the full gene panel on the x-axis and the scaling factors with the skewed gene panel on the y-axis. And cells in the ventricle brain region are shown in green and have a systematically larger scaling factor than other cells shown in gray. And this sort of makes sense because when using a ventricle-specific gene panel, ventricle cells will have higher detected gene counts compared to other cells just by virtue of us measuring ventricle genes that may not be as highly expressed in other cells. And so this tissue-specific bias has corresponding tissue region-specific impacts on downstream differential expression after normalization. And so here I'm showing the negative log 10 p-values with the full gene panel on the x-axis and the negative log 10 p-values of differential expression with the ventricle skewed gene panel on the y-axis. And the ventricle differentially expressed genes are shown in green. And here you can see that they have a much less significant p-value when using the skewed gene panel than when using the full gene panel. In contrast, differentially expressed genes for other regions here shown in gray have a much more significant p-value with the ventricle panel than with the full panel. And this is consistent across count-based normalization methods from library size to SC transform to the 2 bulk sequencing ones. And so this can result in false negatives when looking at differential expression for the ventricle region, as we see here with normalized RIC expression. So on the left, I'm showing library size normalized RIC counts for cells in the ventricle regions compared to other cells. And on the left, I'm showing this for the skewed panel, and on the right, I'm showing it for the full gene panel. And so we see here that there is not a significant overexpression of RIC in ventricles when you use the skewed gene panel, but when you use the full gene panel, there is a small but significant overexpression of RIC in the ventricles. This also results in false positives, as shown here with LGR4 expression in the habenula. And when we quantify these error rates, we find that we can get up to a 4% false negative and a 5% false positive rate, depending on which count-based normalization method we use. We also see a region-specific bias in gene expression fold changes. And so here I'm showing the log2 fold change for genes when using the full gene panel on the x-axis and with the skewed gene panel on the y-axis. And we can see that the ventricle genes shown here in green are shifted down compared to other genes shown in gray. And this is consistent across count-based normalization methods again, and it can result in a switch direction of fold changes when using the different panels. We can see an example of this here, looking at HTR2C library size normalized expression. With the ventricle panel shown on the left, we see that the habenula region has a positive log fold change of HTR2C expression compared to other regions, while with the full panel on the right, we see that it has a negative log fold change of a similar magnitude and p-value. Conversely, looking at GPR75, with the ventricle skewed gene panel, we see that the ventricle has a negative log fold change of about 0.7 compared to other regions. But using the full gene panel, it has a positive log fold change of 0.9. And again, quantifying these switch rates, we find more than 10% switch positive rates and 8% switch negative rates, depending on the count-based normalization method used. When we look at spatially variable gene identification using NNSVG, we find that with count-based methods, there is an overall lower concordance of p-values when looking for spatially variable genes when we use the skewed ventricle gene panel compared to the full gene panel. This can result in false positives and negatives, primarily after count-based normalization methods. And we can see a specific example of this here when we look at the library size normalized PDGFRA expression with the ventricle panel on the left compared to the full panel on the right. If we zoom into the habenular region and look at the data with the ventricle gene panel, you don't really see much of anything. But when you look at it after normalization with the full gene panel, you can see that this region seems to have a higher expression of PDGFRA compared to surrounding regions. And so quantifying this false negative rate, we find up to a 7% rate of false negatives with count-based normalization methods. And we note here that we don't quantify the false positives just because with the full panel, almost all of the genes in the data set are spatially variable. And so overall, these results tend to indicate that count-based normalization methods can lead to region-specific biases in downstream differential expression, gene fold change, and spatially variable gene analysis. And using the same approach, we evaluate these effects in other simulated gene panels with the same MERFISH data set, as well as data from other imaging-based SRT technologies, looking at different tissue types. And so I'll show the data for that really quickly. So looking at 3 other skewed gene panels from the MERFISH data, we find similarly increased rates of false positives and false negatives on differential expression testing. We find similarly increased rates of switch positives and switch negatives on fold change analysis. And we find similarly increased rates of false negatives on spatially variable gene identification, primarily after count-based normalization. These effects also hold true when looking at mouse brain data assayed by a different technology, StarMap+. And it also holds in other tissue types, for example, this mouse kidney assayed by Seekfish, human breast cancer assayed by 10xenium, and human liver assayed by Cosmics. And so what does this mean for our data analysis? So using our simulation approach, we explore a couple of different approaches to mitigate these impacts and make recommendations based on these observations. So first, we go back to the MERFISH data, and we simulate gene panels that are more representative of the overall expression of the tissue. And we find that tissue-specific biases in scaling factors, in differential expression p-values, as well as in gene log fold change analysis, are much less significant than what we observed before, when the gene panel is more representative of the overall gene expression in the tissue. We also use single-cell RNA sequencing data to simulate cell-type skewed gene panels of increasing size. And perform similar analyses to what we did before. And we find that as the gene panel size increases, the cell type specific biases that we observed before are mitigated, both for scaling factors as well as for differential expression, p-values, and gene log fold changes. And so this is likely because as the gene panels get bigger, they become more representative of the constituent cell types. And this is great because as we heard in the previous talk, as imaging-based SRT technologies advance to reliably include more and more genes, the issues presented here will be less impactful on downstream analyses. And so based on all of our results, we present the following decision tree to kind of help us think about what kind of normalization to use in different contexts. And given that the technical factor we're really trying to correct for here is partial volume capture, we think that the ideal normalization factor is volume, as we kind of saw in our very first simulation. However, cell volume estimates can be hard to come by, especially in publicly available data. Also the reliability of cell volume estimates can be susceptible to errors in cell segmentation. However, if you are confident in the cell volume estimates in your data, then we think that normalizing by cell volume is the best option. However, if this is not possible, then that is when an evaluation of how representative the gene panel you're working with might be necessary. And so to do this, we recommend using matched untargeted gene expression data, such as single cell RNA sequencing data from many of the available atlases that are now public. And if your gene panel is fairly representative of the tissue being used, then total counts are likely a reliable surrogate for how much of your cell volume is captured in the imaging. And therefore, using single cell normalization methods like library size normalization or SC transform will likely not lead to significant biases in downstream analysis. However, if your gene panel is not representative of the tissue assayed, then total counts are likely not a reliable surrogate for cell volume captured. And in this case, we would probably recommend restricting subsequent analysis to the region or cell type that the panel was designed to probe. And so overall, this investigation motivates the need for new and specific statistical and computational methods that specifically consider imaging-based SRT technologies. And here we're just examining kind of one step in the analysis pipeline for imaging-based spatial resolved data. And we're really only looking at within experiment comparisons between different cell subpopulations. And so further investigations are definitely needed to examine kind of the reliability of multiple sample experiments. New methods will be needed to enable kind of cross-experiment as well as cross-technology harmonization for us to really be able to leverage kind of the true potential of these emerging technologies. And so with that, I'd like to thank you all for listening and thank my advisor, who's a Gene fan, who's been instrumental in this work, Kaylin Clifton, with whom discussions kind of inspired the initial stages of this work. Kaylin and Manjari developed ST-Align, which enabled some of the MERFISH analyses that I talked about earlier. And Gota Ayahara developed SE-raster, which made the spatially variable gene analysis presented here computationally feasible. And I also want to note that the first version of this work is available on bioRxiv at this link. And yeah, with that, I'd like to thank you all for listening. And yeah, I'll stop sharing.